Hey, biology, this is Mr. Hausman. We're in the middle of our genetics unit and we are developing our skills of being able to predict the outcomes of genetic crosses. That means to try to characterize the genotypes and the phenotypes of the children that are created by reproduction. And what we have been working on was we had several lessons over the past few days, but we're trying to build to gain more independence. And page 3 was assigned early this week on Monday. And then more recently, students were asked to work on pages 4, 5, and 6. And we didn't get as far as we thought on Tuesday, but this video is going to help reassure you on past homework on page 3. And then we're going to build some new skills, and we'll see that on the next page. So we are predicting the probability, right, of traits during genetic crosses. And you have to read each paragraph and you have to do some decoding. You have to feel like you're kind of a forensic scientist or maybe a detective. And so in this first one, number 4, they talk about having fingers on your hand. Now, you can see, right, I have 5 fingers, but believe it or not, having extra fingers, that is being what's called polydactyly. Poly means many, dactyl means fingers. So having 6 fingers is actually dominant over 5. Now remember, dominant means that the allele for 6 fingers will mask the allele for 5 fingers. It doesn't mean dominant in the population. It is not uncommon to meet people who have an extra digit on the outside of their hand. They sometimes have extra digits in the middle, and there can even be extra thumbs. And it has to do with a gene that separates your fingers during your development as an embryo. So, and a few other genes that are involved in bone formation and things like that. So let's decode the information. Let's go to sentence number 2. A man is born with 6 fingers. So the man has 6 fingers. And we know that that's the dominant allele or the dominant trait. The mother, if you keep reading, right, marries a woman with 5 fingers. She has only 5 fingers on her hand, on each hand. And they have kids. But let's slow down for a minute. Let's think about what we know about dominant and recessive. Well, it doesn't tell you what allele to use, but let's use D for digit, not for dominant, but for digit. And capital D is the allele for 6 fingers. You have to kind of pick that letter unless it's given to you. So D for having extra digits and little d for having 5 digits. That's the recessive allele. So if you think about it, because little d is recessive to big D, 5 recessive to 6, the only way to have 5 fingers is to have a genotype of little d, little d. But there's 2 ways, 2 chances, 2 different genotypes you can have to have extra fingers. You could have 2 of the dominant alleles, right? 2 is, pretty dominant. And then we have heterozygous. Heterozygous has 1 copy of that dominant 6-finger allele, but that's all you need with simple dominance. So we can already start to figure out some of the parent's genetic code. And we're going to use blue for that. So the man has 6 fingers. Well, we don't know which genotype he has, but the mom has 5 fingers. So right away, we know that mom must carry in her cells the genotype DD. But due to the law of segregation, we would put one of those D's above this box because some of her eggs are going to have little d, and then the other one goes there. Those are moms. And the parents always go on the outside. I think most of us know that by now. Now, how do we know what to put in for dads on the left side to be able to predict the outcome? Well, here's where we have to be a little bit like an investigator. Let's read the next sentence, okay? They have 4 kids. 2 of the kids are born with 6, so let's write that down. 2 of the offspring have 6 fingers. 2 of the other kids, the other 2, are born with 5 fingers. And that 5-finger combination is actually very important to know, specifically the fact that that they have 5 fingers means that we know these kids, genotypes. The only way to have 5 fingers is to be little d, homozygous recessive. And so notice I'm putting the known children, right, into the Punnett square, not where the parents go, but where the offspring go, the possible offspring. Now, I bet some of you watch crime shows and you know that detectives look at evidence, and this is evidence that we know is true, and we think about what caused that. Well, you know that in this cross pattern, this D came from up here, and that tells you that this D must have come from over here. So the dad's part of the dad's genotype must have contributed a lowercase d because the mom contributed the other one. Now, we also know that there are children with 6 fingers. And you say, well, do I know whether it's big D or big D, little d? Well, let's think about how it must work. Mom contributed, right, little d, and that's the only allele she can contribute. So let's kind of work forward here. So mom can only contribute these guys. So what does dad have to be giving to these 2 kids, the ones in the upper boxes, that gives them the ability to have 6 fingers? And that must be, dun dun dun, a dominant allele for extra fingers. And so there we go. We now finish it, and we realize that dad's genotype had to have been 1 capital D, 1 lowercase. And we know that that's called heterozygous. So is there a ratio of genotypes? Sure. 2 out of 4 are heterozygous, and another 2 out of 4 are homozygous recessive. Some of you know that that's a 50-50 ratio. That's also called a 1-to-1 ratio because 2/4 is equal to 2/4. But that's a 1-to-1 ratio, heterozygous to homozygous recessive. Remember, genotypes are the alleles that the person carries. The phenotype is the appearance or what it looks like. So let's now do the final step. Let's decode. What do these offspring look like? Well, you actually had the phenotypes in the given problem, and it says that it's 2/4 have extra digits, 6 fingers, and 2/4 have 5 fingers. And that is how you use a combination of deductive logic and some analysis of all of this description. Now, it mentions some other things. I didn't ignore them, but the man's mom, had extra fingers. Well, in order for him to have extra fingers, his mother, right, this man's mother, would have had to have been homozygous recessive. Would have also given him, I stand corrected, 5, homozygous recessive. So there we kind of have number 4 done. Number 5 looks harder because it's about characteristics that you don't know, but essentially we have pigeon color, the color of their feathers. Nice try, Hausman. So capital B is the allele for red feathers. It's called ash red, but let's just call it red feathers. Little b is the allele for blue feathers. So capital B is red, and I know that's weird, and lowercase b is blue. And we know that red, because it's a capital letter, is dominant over the blue. B allele, and it says that. So a pigeon with a genotype BB is crossed with a pigeon with a genotype little b, homozygous recessive. And you say, but where's the Punnett square? Well, you know to draw the Punnett square because when there's one trait involved, we always use a 2x2 Punnett square. And we're given the 2 parents, and it doesn't matter which side we put them on, but the parents, the P generation always goes on the outside. And now let's do the simple cross. Let's figure out the probability. And all I'm doing is bringing each letter down. I tend to go the left letters first, but really you always write the dominant allele first, if there is one. So little b and little b, homozygous recessive. And we have actually a very similar pattern to number 4 above. That's a coincidence, right? That it's the same pattern, just different letters. But the question here is complete a Punnett square and then prove that you can figure out what percent of the offspring are red. And that's where red. Hello. The word is red, but the allele is B. So I can look in this table and I can see that the top half, right, the 50% on the top, 2 out of 4, are red. That's its phenotype. And then the 50% are blue. So the answer is C, and your evidence is that it's 2/4 B. And that would be red feathered, uh, ash red feathered pigeons. So that one was kind of easy once you realized you had to draw your own Punnett square. Lastly, let's look at a plant. Tobacco plants. Tobacco plants grow all over the world, and of course, we know that what they make. They make tobacco that's used, in different cultures and things like that. Tobacco is actually grown in New England, in parts of Connecticut. I actually used to work on a tobacco farm. Story for another day. Well, the leaves are huge. They're like the length of from my fingertips to my elbow when they are mature, and they are green. Like most plants are green, right, guys? Now, apparently, there's an allele for being an albino. I've never seen that. That would not be a very successful plant because it can't do photosynthesis. So it must get its sugars another way. So it's unable to. So what color do we use, or what allele do we use? You have to make up your own. But you know that green, it doesn't say that explicitly, but green is dominant over albino. So let's use capital G, green. Now, what does that mean an albino is? I'm writing it over here because I'm out of room on the left side. Albino must be little g. Okay, so we have big G for green, little g for albino because dominant over recessive. And so let's talk about the cross. A tobacco plant that's heterozygous for normal color. So it could have been any letter, but for heterozygous means that one plant has to be capital G, lowercase g, heterozygous. Hetero means 2 different alleles. Now, here's something that you've never seen. If you self-pollinate as a plant, that means the pollen from one plant falls into the female parts of the same plant. So the plant has the same genotype in all cells, so it's crossing with itself. So this is what we mean by a self-pollination. Capital G, lowercase g is the tobacco plant's genotype, and it's crossing with itself. And it says it makes 500 seedlings, and they germinate. That's the next generation. So that tells us something about F1. And here's the parents, of course, on the outside, blue. So let's do our cross. Capital G, capital G, capital G, lowercase, capital G, lowercase, lowercase g, lowercase g. I see something interesting here. I see 3 different genotypes. I see, and I'm going to use some shading colors, I see homozygous dominant, capital G, capital G. I see heterozygous for 2 of the 4 boxes, and I know it's hard to shade over them. It kind of makes them look really different. And then homozygous recessive down here in the corner. So it's actually called a 1 to 2 to 1 ratio, right? Because it's 1/4 to 2/4 to 1/4. Now, what do we mean by that? Well, there's 1/4 are homozygous dominant, 1/4 recessive, and heterozygous in the middle, 1 to 2 to 1. Now, here's the fun part. There are 500 seedlings, right? There are 500 plants that are grown from this cross. And we now are going to use these fractions to calculate how many there would be. How many offspring would have the same genotype as the parent? Well, that's easy. The parent is heterozygous. Here's the parent. So do we see any of the same genotypes? Yeah, 2/4 or 1/2. So 1/2 times 500 is 250. How many would be white? Where's the white phenotype in of these patterns? Oh, I remember that white or albino is the recessive allele. Here's the albinos, and it's only 1/4 of the population. So 1/4 times 500. That's how you take 1/4 of the total number. And that may not be something you can do in your head, and that's okay. But you divide 500 by 4, and you get 125. So a little trickier, and a little bit kind of more interesting to have to do it like almost like an investigator. So these are old ideas. These are based on the pattern of dominant and recessive alleles. The video is going to continue, and it's going to now talk about stuff that we're doing new. We're going to talk about incomplete dominance. Incomplete dominance is when there are two alleles that are both capital letters. So in this example, we have fish, and red and blue are incomplete dominant. So we're going to call one R. I do have a red pen. One R and one blue. But when you have the heterozygous combination, when you have one of each, it's not that red is dominant over blue or blue is dominant over red. Heterozygous appears as a blending of the two traits, not one or the other. And most of us know that if in the world of pigments, although it's very for a different reason, these fish, when they mix their red and blue genes, they get a purple colored fish. So the red genotype is capital R, capital R. That's the only way to be pure red, right? Because if you have any blue, it's gonna mix. And blue has to be pure blue. And then purple has to be RB. And you can write it either way. You could write RB or BR. Okay, so let's read what the parents are. So they first tell you what the trait is. Now it says what would be the genotype and phenotype of fishlets. I guess that's a baby fish. But when there's a red and blue fish crossed. Oh, so you mean that one of the fish, the parents, is red and one of them is blue. And it doesn't matter who's red and blue. But we need to put each parent in around the Punnett square to predict what they might have for offspring. And I'm going to use orange. I know that's weird, but I've been using that for the offspring. No, let's actually, I'd take that back. We're going to use the colors of each allele that I've been using. Fading fast, right, guys? Fading fast. You can see that R disappearing. So we take another red pen. And blue is a little stronger. And now you notice that it is 4 out of 4 RB. Every single offspring for pure blue crossed with a pure red, you're going to get purple fish. And it's going to be 4 out of 4 or 100% purple appearance, because every single box, right, is incomplete dominance, right? So it's 4 out of 4 have a red and a blue allele. So we see purple. So that's new. And anytime you're dealing with incomplete dominance, you have to pick two different letters, make them both capital. And usually what I do is I make a letter that's for the two dominant colors. So let's go to another one. Let's go to guinea pig fur. Guinea pig fur, on the other hand, is another incomplete dominance trait, but we have black and white. You know, good luck writing with white, but let's use B for black. And I'm going to use a pencil for white. I know they're both dark, but that'll be capital W. So dark black, B for black, and W for white. Well, how do you get gray, like in the world of pigments? You mix some black with some white. So let's now see if we get it with incomplete dominance. To be pure black, the only way to be black is to be black fur is capital B, The only way that's supposed to be with a black pen, my fault. The only way to have white fur is capital W, capital W. And therefore, the only way to be gray is a mixture, incomplete dominance of black and white. Now, that's not the parents, that's just the three possible genotypes with incomplete dominance when there are two alleles. So, are there parents involved? Yeah, let's go to the next sentence. A black guinea pig. Aha. So the one parent is got to be capital B, capital B. And you probably assume that they're going to cross it with white. Nope. Read the details. They cross it with a gray. The two parents are BB and BW. And so now when it's time to do your cross, right, the black parent contributes all black alleles, but the gray guinea pig parent contributes 50% white alleles due to the law of segregation, right? It's either B or W. So in the offspring, we have a 2-to-4 ratio of BB and a 2-to-4, 2 of 4 are BW. It's a 50-50 chance of getting, what do they look like? It would be 2 out of 4 are black and 2 out of 4 are gray. And if you think about that, you say, well, that makes sense. The dad or the mom was black, and the other guinea pig was white. So shouldn't they have like a 50-50 ratio? That's not always true. There are other ways that it can happen, but with incomplete dominance, you can predict a new pattern. So instead of a 1-to-3 to 1-to-2 to 1 ratio, you have a 50-50 ratio or 1-to-1 ratio. Okay. Now, I want you to set yourself free and see if you can't figure out using the same animal model where B is for black and W is for white and BW is for gray. What if two gray guinea pigs cross? So hit pause on the video, show the parents' genotypes, put them in the outside of the table, and check your work in just a minute. But hit pause so you can do it on your own. Let's build confidence. Let's build independence. So if you hit pause and did some work, that's great. Now we're going to simply go in and show you the answers. We've got black and white. So that's gray, right? That's a gray parent and a gray parent. So BW, BW, capital B, capital B. And now we're back to that 1-to-2 to 1 ratio. 1/4 are black, 2/4 are gray, but let's put in the code for gray, BW. And then I see down here in the corner, 1/4 are white. So, by the way, did you notice I actually said their phenotypes? That was actually wrong. 1/4 is homozygous dominant. 2/4 are incomplete dominant, right, having both alleles. And 1/4 is, and again, you can't say recessive, but 1/4 are big W, big W. There is no name for the combinations for incomplete dominance. You really just have to list their genotypes. Now, the phenotypes, that's what I already mentioned. 1/4 black, 2/4 gray, and 1/4 white. And again, because they have common denominators, you'll sometimes see me write that as a 1-to-2 to 1 ratio. 1 black to 2 gray to 1 white. And that's a very common way of expressing a monohybrid cross results.